Welcome to Next Big Future, where we talk about big problems and big solutions to get us to a better future faster. Today, we talk about one of the biggest problems, aging, where 150,000 deaths per day are caused by aging, which is a huge number. Just to give it a bit of scale, it's 10 times worse than the worst days of the COVID pandemic, and it causes 2 out of every 3 deaths. So a big solution might be coming from my guest, CEO Daniel Oliver, who has a company, Rejuvenate Bio, and looking at first saving our pets, and then we learn and prove how to save people from aging. We'll go over big new developments with this exciting company, related science advances, and talk about the field of anti-aging and how and when this all happens. My first question to my guest, Daniel Oliver, is to tell listeners new to anti-aging about himself and his co-founder, George Church, and highlights of his company. Go ahead, Daniel.
Yeah, well, thanks so much for having me. Yeah, we have a pretty unique story of how the company started. Actually goes back to my time at Caltech, where I met my the third co-founder and current chief scientific officer of Rejuvenate Bio, Noah Davidson. So we were friends at Caltech. Ironically, neither of us were biology undergrad majors. I actually was mechanical engineering and business, and Noah was an applied physics major. And so coming out, we both, I think, were interested in utilizing science to make a difference in the world. Took fairly disparate paths. I actually went and worked in aerospace, launched a company around creating better wheelchairs for developing countries, and got a real appreciation that it took quite a bit more than scientific knowledge and engineering knowledge to actually make a difference, even with a really key scientific innovation. And so I got drawn back to school, ended up getting my MBA from Harvard Business School, was lucky enough to be awarded a fellowship coming out of the MBA program, the Blavatnik Fellowship, that actually paid me for a year to try to commercialize science out in one of the labs around the university. Ended up spinning out a novel 3D printing company. Got a lot of great experience with trying to translate science out of a university lab. And in the background, of course, had still been friends with Noah, who had subsequently went and got his PhD in synthetic biology at MIT, and then joined George Church's lab, interested in working on aging and age-related disease. And what Noah started realizing was there had been a whole host of knowledge developed around interventions, particularly in mice, that had shown an ability to extend maximal lifespan, somewhere between 15 to 30%. So basically 65 different genetic mutations have been tested in transgenic mice, meaning they had bred the mice to either have a gene upregulated, meaning turned on more, or downregulated. And about 6 months into his postdoc, Noah got a puppy. So Bear, when I met him, he's a German Shepherd, but he was about this big. He subsequently grew to over 100 pounds and was, his head was bigger than he was when I first met him. And I think that was a real seminal moment for Noah, because the question came into his mind: why can't we utilize all of the, discoveries we've seen in mice to actually make a difference in species that, let's be blunt, people actually care about, whether that's another human or a dog. And so this is where this idea of re-regulating key proteins in these patients came from. And Noah came up with the idea of utilizing these genes that have been discovered in these transgenic experiments to package them in a gene therapy, which in some sense is just a way of transferring genetic material to utilize to create initially gene therapies that have shown an ability to treat multiple age-related conditions. And then long-term to continue to build on that to actually drive towards what we call age reversal. So that's the genesis of the company. George was obviously there hand in hand with Noah on all that research that was developed. And then also, as I came in as an entrepreneur in residence to help spin the company out, George was also very involved there as well.
And George has had many other companies he's created before, right? He at Harvard, and he created many other companies. I'm not sure if we kind of went over.
He has a world famous conflict slide, if you've ever seen that. I think you can Google it and find it, but it's like, he's got this conflict slide, and I think it's got like over 100 names on it. He basically should have a slide that says like what company's not conflicted with. I think it would be shorter. So yeah.
Right. So some recent new developments is that you've gotten $10 million in funding on top of, I think, about $7 million before. right?
Yeah,
And
we've from different sources, we've raised over $30 million into the company thus far. And our past fundraising round was actually close to 14 million. Yeah.
okay. And you are currently working on, at least on your website, talks about the work with the King Charles Spaniels and their heart disease problem. And so you have a study with Tufts that is looking to address that. Can you describe that a bit?
Yeah, so one of the key things we discovered early on, obviously this company was born out of a passion for dogs, man's best friend. What we started realizing when we started looking into all the regulatory concerns and the development paths it would take to actually bring therapeutics out that would have a benefit for multiple age-related diseases and eventually for aging generally, there was a couple of key considerations. One, no matter how efficacious your therapy would be for treating, something more general like aging, the FDA is going to view it and grade it as if it's a treatment for a single indication that goes through a very standard clinical trial and does the kind of very typical balance of how efficacious is it versus its safety profile. So that drove us to first off take these therapies that we think will have general benefits, but direct them at very specific indications that we can prove in a reasonable time frame are having an effect for an age-related disease. The second portion we started realizing is to answer some of these larger questions that I think everyone's really intrigued by and interested in. Working in humans, even if you were allowed to work in them without any regulatory considerations, is a really difficult patient population to deal with. The timelines of the types of things we're measuring are possibly decades long. Dogs, what we started realizing, were a really great model for human aging and also happen to be a really great model for gene therapy. And so actually a number of the early gene therapies that have been approved, including Luxturna, and also Spark Therapeutics, the first gene therapy company to get approval by the FDA, for AAV-based gene therapy, I should say, actually use dogs as their large animal preclinical model for their first 2 programs. And so a lot of empirical data out there suggestive that what happens in dogs with gene therapy is what happens in humans. Couple that with dogs suffer from a similar list of age-related conditions as humans and also share our environment, so share a number of kind of exogenous factors that could drive problems. You have a really interesting target population to get a treatment into to see what would happen long-term in humans. And what we started to understand is there were large challenges in animal health that we could go after and conquer. And I'll go talk about that trial in a sec. I know that's what you asked about. But also getting our therapy into animals would be a really key step, and I think necessary step, for actually proving some of these long-term theses around, are we increasing general health? Are we preventing other age-related disease? Are you seeing an increase in maximal lifespan eventually? All of these questions are much easier to answer in dogs. And so we ended up developing a program where we are able to utilize what typically would be preclinical data to get an IND to launch a human clinical trial. We're able to get a lot of crossover there and actually launch an animal health product. And we're targeting our first therapy against the treatment of the leading type of heart failure in dogs called mitral valve disease. And you mentioned the study in Cavalier King Charles Spaniels. The study is actually sponsored by the leading breed club in the US for Cavaliers, the American Cavalier King Charles Spaniel Club. We're running it out of Tufts Veterinary School, one of the leading veterinary schools in the country. And in the study, they enroll dogs who are presenting with this type of heart failure called mitral valve disease. The dogs are then given the gene therapy and then monitored. And we've actually had dogs on the gene therapy now for over 2 years. So really exciting. Study is still ongoing, so I can't release specifics about the data, but I'll say we're very encouraged by what we've seen. And we think it's a really novel way of developing therapeutics, particularly in this area, meaning gene therapy and aging, by utilizing animal health as both a place where you can solve large challenges and then also utilize that data to help inform what you're doing on the human side. So we always actually talk about doing it in parallel. And it's funny, people ask us like, oh, is this experiment for dogs or humans? It's like, it's always both. That data is very, very useful. And why not try to use it in 2 places?
So just to clarify for people, IND is an investigational drug study. So that was the IND you're trying to get, the approval is an IND for dogs, not for humans, right?
So we have an open INAD
Mhm.
for animals. And so, yes, you're right. An INAD or an IND is the necessary regulatory step before launching a clinical trial. You basically go to the agency, say, hey, this is what I would like to do. It's a little more complicated than this, but in general, you go to the agency, the FDA, go, hey, this is what I would like to do. This is the data I have to support what I'm doing is worth trying. And then they sign off on your plan, obviously with some edits.
Right.
And then you're able to go off and start those studies. And whether it's an INAD or an IND, similar. What's interesting is you have to do a lot of work in animals to get an IND to launch a human clinical trial. And the overlap between what you would have to do to launch a product commercially, so to get through the whole regulatory process in animal health, has a huge overlap with what you would have to do anyways to launch a clinical trial in humans. And so in some sense, those things are like kind of one-to-one. Not exactly, but close.
Great. So so that means since you've done the 2-year trial and they overlap with the human work to get the approvals on that, is that something that you feel close on? Is that something that we could see beginning of a human trial? Because in your pipeline, you have, I think, 3 genes you're looking to target in humans, and you have the application in for the IND. So that approval could happen later this year, beginning next year. What's your general feeling on what's happening?
Yeah, we are getting closer. I'm not going to give a specific date because you know how these things go, but I will say that we're getting close enough that we signed a commercial relationship that hands off commercial responsibilities for our animal health product to a leading animal health company that is excited enough about what we're doing to sign a deal with us. I think that's awesome. It'll ensure that we can get the therapy to all the dogs who need it. And then allow us to continue focus on early stage R&D and clinical development, particularly on the human side. And so kind of subsequently with getting to commercialization in animal health, we also are planning to basically launch a human clinical trial with our first therapy. And to be clear, you mentioned 3 genes we're experimenting with, which is correct. Our first gene therapy, RJB01, will go into commercialization in animal health. And then a humanized version of it, we actually recode the genes for the specific species, would be going into a clinical trial in a similar time frame. And so, yes, we're rapidly approaching commercialization in animal health and, hitting the clinic for human health. So those things are very much on our near-term horizon.
So let me go over the animal thing. So it's Phibro or Fibro
Animal health, yes.
Animal health, right. So that's the deal. So they would take over the implementation or the work with that you're saying there would be more of a handoff.
The commercial aspects. So, Rejuvenate handles basically to approval. And then Fibro is an expert at commercializing animal health assets and have the resource to do so and would take over those types of responsibilities. And so it's a nice marriage where we can provide the technical knowledge needed to kind of get to the finish line clinically. And then Fibro can utilize their commercialization knowledge. And we don't have to develop a commercial organization at Rejuvenate Bio.
Right. So then, and then you might do this model in order to get to other places around the world where if they didn't have enough coverage globally, then you could say, okay, another company could help get it done in Europe or Asia or whatever, right? If they didn't have the coverage for that.
Correct. This would work across pan geographies.
Right. And then in terms of how this gene therapy works in dogs, in terms of the actual treatment of it. So you use the viral method to carry the gene therapy into the dog, and what it.
Do you want me to just walk through it kind of step by step?
Why I should walk through
Yeah. So like, I'm an engineer by training. And so I actually think gene therapy is really intriguing because in some sense, although it's like the novel, treatment method, I think it's a little more direct than even what's happening when you inject a typical drug or small molecule, as they're called. And so with gene therapy, you're using a specific type of virus called adeno-associated virus. And this is interesting. You basically take that virus, rip out its internal machinery, And so that stops it from replicating. So it's a non-replicating virus. And then you basically put in your gene of interest. In this case, it's 2 different genes, and then some other machinery that allows those genes to be turned on once they get into the cell. You then create a vial of this virus. We administer as a one-time intravenous injection. So it's actually, in administration, is fairly simple. This particular strain of adeno-associated virus is what they call liver trophic, meaning it typically infects liver tissue. And so it goes in, these viral engineered viruses drop off your genetic payload, which in this case is a couple of rings of DNA, or called cDNAs. Those rings of DNA sit next to the chromosomes in your cells. Don't change the underlying genetic code, so there's no editing happening. But they sit there, and then they go through that. If you remember high school biology, DNA goes to RNA, goes to protein. And so they basically use your cell's machinery to create protein. That protein, in our case, is what they call a secreted protein, which is just what it sounds like. It's secreted out of the cell, goes into the bloodstream, and then what's really having our therapeutic effect is that secreted protein. So in some sense, if you think of that whole string, we're using a virus to basically get a higher level of a certain protein in your bloodstream, which then causes an effect widely around the body. The interesting part about this is it's a natural occurring protein. And so you're not kind of shooting blind with what it's going to do in this part of the body or that part of the body. We know quite a bit about what it's going to do. And the fact that we sourced our targets from these transgenic longevity experiments in mice, where they had this intervention in every single cell in these animals' bodies, we are very confident that at least our pedigree is that these treatments would be incredibly safe to do that. And we thus far empirically have also seen that when we've injected a number of animals with them, including mice and dogs. And so that's how it works. I think it's, again, being an engineer, it's, I think, interesting now that we're at the part of biology where we can repurpose specific tools there to create therapies to accomplish goals we want. And all of these kind of genetic therapies, in some sense, are that. It's like recapitulation of a number of different tool sets to create a therapy that accomplishes a goal you want to do.
So in terms of you getting the genes to the cell, but not changing the DNA, and then they're producing protein, so then that would be for treatments where there's a lack or insufficiency of that protein, right? And not for something where
like downregulating, you're saying would be hard.
Yeah, downregulating would be a different approach, or you can address that maybe in some other way.
So actually so you are spot on, and that is a great question. So one of our proteins, exactly what you said, FGF21, we're trying to upregulate it, and it's just what I talked about. The second, the second gene we're encoding for is actually a receptor that binds a circulating protein and inactivates it. And so in some sense, that is how we get down regulation, is we overexpress a receptor that then floats around your bloodstream, binds to the gene or the protein we're interested in activating, which is TGF beta 1, and basically inactivates in the system. And so that's a method of down regulation you can have from, as what you point out, is a more logical way of upregulating a certain protein. Okay.
And so it's genes, RNA, and proteins. So there's been a lot of huge work, recent developments in with DeepMind, AlphaFold kind of solving proteins. And then there's Perturb-seq, I guess, where they were able to map. So basically these two kind of lab science results, where basically we've made a lot of progress figuring out what the proteins, and they're going from sequence to folding. And then we've had, which was the DeepMind work with AI, not complete solution, but it's something that is really rapidly advancing the field. And then the other area is where I think it'd be the MIT group has been using a new technique to kind of like map genes to functions. Do you want to address either of those things and how those kind of combine to make the science in your area perhaps a lot faster?
Again, both are really cool tools. In particular, the ability to read out what the folding of a protein will be and what the structure is starts really helping to inform like what the interaction would be with that protein structure. And so not as directly relevant for what we're working on right now, at least as a therapy, but like behind the scenes, there's definitely some work there that could be very relevant for us. And then the question of mapping like genes to function, you know, I think is like an ongoing area of study that always is very useful. So we think about trying to utilize signaling proteins that have large cascades that obviously affect multiple different, gene networks, because in some sense, if you're really trying to attack aging, you would imagine you need to attack more than one issue to basically push that horizon out. And so obviously knowing, what these specific genes being upregulated or downregulated do as far as function goes is super important. It's also some of the work we've done to categorize what downregulating or upregulating these genes, both in singles and combinations, do as well. And so one of the key elements or kind of key academic literature that came out of Noah's postdoc with George, that was a foundational piece of our starting the company, was that when we took these two genes that became RJB01, we were able to treat 4 different models of age-related disease in rodents. So a model of heart failure, model of kidney failure, induced diabetes, and obesity in mice with the same treatment. And so in some sense, exactly what you're talking about, like we're mapping what happens when you upregulate or downregulate these proteins in singles and combination. But in this case, not to necessarily like everyday function, but how it would affect a specific aging process. And in this case, a model of age-related disease. So all of that stuff is very useful. And I view biology right now as like aerospace, like post-World War II, where they're breaking the sound barrier, re-breaking it, pushing jet technology forward. And like every day you're seeing like another huge leap in different areas. And in some sense, all of these things are additive for an overall picture.
So in terms of the path forward for Rejuvenate Bio, it's getting into more and more breeds of dogs, and in parallel, getting more different treatments, different genes, different gene networks treatments into humans, and kind of doing those in parallel, right?
Yes. So 2 things there. So we have been in multiple breeds of dogs. So while the Cavalier King Charles Spaniel Club sponsored this research, it actually has extended to multiple breeds in this trial that I spoke about. So that's been awesome. The treatment is not breed specific, although unfortunately Cavaliers do have very high prevalences of mitral valve disease. And so as you can imagine, their pet parents are very aware of this issue and are very motivated for looking for treatments. The next step for Rejuvenate Bio, I'll just take a step back here. We view aging as a problem of gene expression. So the DNA you have the day you're born is effectively the same DNA the day you die. Maybe you have some point mutations because you were too much out in the sun or something like that, but overall, that blueprint is pretty much the same. What really changes through time is which genes are being turned on and off, and so which genes are being expressed. And there's quite a bit of data out there outside of our, outside of what Rejuvenate Bio's developed, that shows if you're able to change that gene expression profile, basically the relative expression levels of the genes in your body to what it was when you were, pick your favorite age here, 25, 28, that system then starts working more akin to what it did when you were 25 or 28, where you don't see, you typically don't see high rates of cancer or heart failure or stroke or any of these kind of what we classically would view as age-related conditions. And so with our first gene therapies, we're really taking 2 different proteins, one that goes up through age and down-regulated, one that goes down through age and up-regulated, and have already shown that even doing 2 of those has an effect in multiple age-related conditions. We've shown that both in rodents and large animals, in our case, dogs. What we're interested in doing next, and we have proof of concept data and have ongoing studies, although very more research-based now, is utilizing something called epigenetic reprogramming. to not affect one or two genes at once, but literally thousands at once. And so epigenetic reprogramming really holds this power of changing gene expression profiles across the board. And so you could basically change the system to operate as it did, where you could call it younger, but we like to say when you were healthier. And that's where we really see, that's where you get into these actual talks about age reversal. And we've been able to produce some things internally that make us very bullish on that, I can't discuss quite yet. They're getting close to publication, but really exciting stuff there that could really push the envelope past, just one or two re-regulations, but literally pan your entire genome.
So just to talk in general about epigenetics and so that's the methylation. So basically there's an attachment to the gene that turns it on or off, which is the methylation part.
Exactly.
And there is a, I think a Horvath clock or something like that related to epigenetic age.
Time to death is, I think, the best way to describe that. Time to death.
It is time to death. Okay.
And so, so instead of like talking about your chronological age, the Horvath clock is good at pointing out to like how much time you have left, which in some sense is the metric you really want. Yes.
And it also goes back to the beginning. So basically then it's how good is this clock in terms of like the precision that you need, like 1,000 different things that you're changing. So you'd want to have the research would been done to kind of like research out like, or just look at old age versus that 2025 ideal age kind of thing, and just look at the differences, right? And then use some AI or some large data analysis to kind of like say up, down, up, down, up, down, and just kind of like say, here's what we want to get it back to kind of thing. Is that how that would kind of work, would apply?
So the Horvath clocks or the epigenetic or the methylation based clocks, I think are really promising as like surrogate biomarkers, such that long-term people wouldn't have to run like full longevity studies. In some sense, they're not, in my opinion, they're not there yet. They're certainly not accepted by the FDA as like a relevant metric if you were trying to get a therapy out for, through the regulatory process. And so we are taking a very similar approach where we're, long-term would be taking these therapies that we think would have broad applicability, but again, taking a very scientific, data-driven approach to, discipline ourselves to look at specific indications, measure the impact on that specific indication, and then continue to build up our knowledge and basically our reach by going into ever-increasing larger patient populations. And so that is the way we think about doing it. Yeah.
So there's the TAME study, which is I think the first study for the FDA that was treating aging as kind of a disease. Isn't that what that study was trying to achieve, or whether
it's the metformin one you're talking about?
Yeah, the metformin study. Yeah.
Yeah. I think even that they're looking at, and this, maybe this will be an area we can review, but my understanding there is even that they're looking at diabetes endpoints on that.
Okay.
And then again, that is like a more academically driven study, is my understanding too.
Okay. So in terms of indications, then there have been anti-aging groups, enthusiasts, people who are doing that, who have go through a long list of aging tests, where basically how many push-ups, how many balance, and then do blood studies.
Health span type stuff. Yeah,
health span type stuff. Is any of that kind of like, if you want to get a comprehensive indication thing, or basically you have to go at these specific diseases, have these indications that FDA approves, and you can't go to, your biological age kind of seems to be whatever, right? It has to be more the FDA.
So I think two things to think about, we think about both, and we think both are important. Like, there's a lot of things about if you could create an age reversal therapeutic that would be very additive for our health system, So the idea here would be, instead of a laundry list of negative side effects on your treatment, you'd have a laundry list of positive benefits of what you're doing. So it is important to look at things like that, like quality of life metrics, like you said, basically like ability for maximal exercise, ability to recover from a wound, like more plasticity in your body so you're able to regenerate and recover from stress, these types of things, all things that I think would be put under the umbrella of health span, and quality of life.
Yeah.
The FDA is not set up to really look at stuff like that. They are far more set up to look at what I've described. We have an indication that's defined. We have a treatment for that indication, and we are going to view and weigh the positive benefits of your treatment versus the safety of your treatment. And you see this borne out with like chemotherapy drugs, which if you or I just took are basically poison. But obviously, if you have a certain mutation of cancer, a certain type of cancer, are the only thing that could possibly save you. With our therapeutics focused in the aging field, I think what we have going for us is we have things that, as I've mentioned, particularly what we're working on, have pedigrees that they should be very safe. Empirically, they've shown to be very safe. And we are continuously generating data that I think they'll have high levels of efficacy of treating specific age-related diseases. And then we can start going into more and more prevalent age-related diseases to try to get larger swaths of the population long-term. Now, the way of circumventing all of this would be, as you brought up, if there was a surrogate biomarker, something that we could track, that everybody agreed that, hey, if you change the signal, you've made the patient healthier, you've made them live longer. And then we could just measure that and see if we've changed that. But there's nothing like that that's accepted now. I think, this is my personal opinion, I think the Hovarth clocks or the methylation-based clocks are the closest thing we've seen to something like that. But they're still not there, and they certainly aren't there from an acceptance by a regulatory body. So I think in the near term and middle term, very key for any company who's operating in this space to be disciplined to picking specific indications that they can, go and measure their impact with their therapy. And then if they do want to answer these questions long term, they need to be operating in a way that they can generate that type of data in a reasonable time frame. And again, this is why we view animal health as a really key aspect of our strategy. Because in some sense, it circumvents a lot of these issues. We're able to get into a large amount of animals, solve a huge problem like mitral valve disease, leading type of heart failure in dogs. About 7% of all dogs get it. Get it into those patients, follow them, and a dog's lifespan, unfortunately, is about an order of magnitude less than a human. And so subsequently, all those things that took decades in humans take years in dogs to measure. And we can start generating literally tens of thousands of N values, or tens of thousands of dogs on the treatment to start answering some of those questions and circumventing some of those issues you're bringing up. Now, am I excited people are working on those surrogate biomarkers and hope they come through? Absolutely. But until those things do, I think we need to utilize strategies like we are to answer some of those really important questions that you've brought up.
So do you think that certain age-related, more age-prevalent conditions, arthritis, immune system issues, the immune system problem was a huge thing for the COVID pandemic because the people who were older were, 10, 20 times more.
Yeah, it was like age and obesity were the biggest ones, right?
Biggest obesity, right. So are those something that will end up being addressed, possibly solved first, one because it's something more limited perhaps, or is it something that is kind of like more likely to be the whole thing because of the 1,000 conditions that you can't really get even at these seemingly more separate conditions without hitting kind of like the whole thing?
Well, really interesting you asked that question. So I'll remark on a couple of things and try to sum it all up together. But first off, upregulating FGF21 is shown to increase your immune system function. And so makes sense if having a heightened immune system later in age would be beneficial. Something that was able to increase your lifespan should, do something like that. So that directly is there. You also mentioned with COVID that, obesity was a large issue. FGF21 also basically eliminates excess fat stores as well. And so what we've seen in rodents in particular is that we are basically able to take them back to their lean body weight, eliminate excess fat stores there. So I think for something like COVID or an infectious disease, which you wouldn't necessarily think aging treatments would have a lot to do, because that's just something you either get or you don't. I do think it would be very beneficial. And as again, this goes to this idea of generally increasing how the system is operating. You know, typically younger folks don't die from infectious disease. They're not dying of the flu every year, as you know, lots of older folks are, and we saw that with COVID as well. How do we get into indications that are more prevalent? We do have data in rodents showing our second therapy, RJBO2, was efficacious in treating a model of osteoarthritis in rats. But for our first therapy on the human side, we actually are initially selecting an indication in the cardiac space and one in the metabolic space, both based upon the literature available. We think we have very high probability of clinical success. Both the indications are rare orphan diseases, so we'll be able to run a smaller clinical trial quicker. And then the idea there is that we are, very hopeful at targeting and solving those problems in those two patient populations, which unfortunately have low standard of care. But also the data we generate in each of those indications will give us first in human safety data showing that our therapy is safe, but also produce efficacy data that is really relevant for follow-on indications in both cardiac health and metabolic health. And so this is key because we can run a relatively short, relatively small human clinical trial and get proof of concept data to then continue to launch our therapy into follow-on areas. And the key here is with that is because we are not utilizing gene therapy to do some sort of gene replacement, which has traditionally been used in for monogenic diseases where there's kind of a point mutation that you're trying to solve. We are bringing an upregulation of certain proteins that create benefits for the patients that are able to treat the kind of downstream effects of these disease. And for instance, in the cardiac space, we're going to something called arrhythmogenic right ventricular cardiomyopathy. It's a mouthful. ARVC is what it's colloquially called. The interesting thing there is we'll be able to look at endpoints that would be suggestive of also going into follow-on cardiomyopathies based upon the data generated in ARVC, because we're not attacking a specific genetic subpopulation which has a genetic variant that causes ARVC. That makes sense. And similar story on the metabolic side as well. And that's how you start driving it into initially patient populations that are in the thousands, then to the hundreds of thousands, to the millions, tens of millions, and then beyond. Inside of our current regulatory framework without having to change any of that or being reliant upon any sort of surrogate biomarker or any change in the FDA on how they view age reversal or anything like that going forward.
Would that eventually, would your work eventually lead to the damage repair thing that from SENS? Like they have the 7 types of damage, and then their argument is that you look less at metabolism and try to slow that down, and kind of like repairing your car is I have rust, and before the hole develops, I'm fixing the rust damage and doing that kind of thing. So basically, fixing accumulated damage.
Senescent cells in that whole area.
Right. And that's something that
lots of different ways to look at aging. I think if you were to look at a 25-year-old, they have less senescent cells.
Right.
So the clearing functions, the production of senescent cells are just less. And so is clearing senescent cells something that could be beneficial? Like, absolutely. Do I think that's the entire answer to the question? No. Do I think you're probably better off focusing on re-regulating the system to when there were less senescent cells? Like, yes. Does that mean like coupling that with like an anti-senescent regime or something like that would not be helpful? Absolutely not. Very well could be something that could be additive to what we're doing as well. But again, I think the key step is really this idea of returning the system to how it was operating when you were healthier. And if you're able to do that, a number of these different aging pathways that people have focused on and done lots of really great work in some sense, you get the benefit of all of them if the system's operating as it did when you were younger or healthier.
Mm-hmm. In terms of the whole field of anti-aging, there's a list of by Kevin Carl Flager, which has a list of like 158 companies working toward anti-aging, and they've had about $10 billion of funding and $7 billion of IPO from valuation from 13 companies or 17 companies like that. So do you see, in terms of you look at the field, do you see certain things happening in terms of certain players getting to a point where you think maybe they'll trigger something like the mRNA boom kind of thing? Is that something that you see happening, or is that too speculative that you don't really think around those lines?
I think there's always two things that need to happen with those types of booms. Like, one is the technology needs to be ready. And I'm happy to say there's lots of really smart people, and I don't consider myself one of these folks. Let's be clear, I'm not an inventor of the technology, but lots of really smart people working on a number of different aspects, some of which you brought up today, on a daily basis, pushing these things forward. The second thing is some sort of event or set of circumstances that makes it a necessity for these types of technology. One of the things we point out is, if you look at overall US healthcare spending, we spend basically $1.5 trillion a year in healthcare spending that I think could be classified as due to age. And so the major way I do that is you look at what we spend on people 65 and above versus people like 20 and below. It's like $1.5 trillion difference, right? So if you look at what's happened to our GDP percentage of what we spend on healthcare because of those types of trends, we've gone from, over the course of the last 40 years, from like 5% of our GDP spending on healthcare to now 20%. And so the question becomes, can we spend 40% of our GDP on healthcare? Like, what is the number where it becomes untenable for us to keep giving the current treatment regimes to the patients we have? And so to me, we are increasingly coming, and you see this in the healthcare debate, and I think everybody would agree that our current healthcare system has gigantic problems. But what if you were able to deliver therapeutics that more return people to these age when people don't want to get health insurance because they don't ever get sick, and these types of things? And so I think in some sense, this is the promise of age reversal therapeutics, is that you are creating a healthier patient population going forward. And so do I see there's going to be an mRNA boom because we have a once in 100 years pandemic there? I don't know if we'll have a seminal event like that. That seems unlikely to me. But I do see that our healthcare spending at the United States, and I think everybody can agree, is kind of an out of control freight train that is eventually going to smash into something. And they are in dire need of therapeutics that don't just kind of patch over issues and create other issues, but holistically are increasing the health of our patients moving forward. And so I think when these therapies get out there and are moving, and I'm not just talking about what we're doing, but this idea in general, I think they're going to be, uptaken and be incredibly embraced, because they're going to be solving not just a specific issue for a patient. They're going to be solving holistically the health of the patient, is the promise, and then also solving large problems for our healthcare system as well. And so there will be a financial and economic benefit of scaling these technologies, or the reach of these technologies going forward. So I'm very hopeful there. There's a number of things going on that I think are very promising out there, including what we're doing. Obviously, I'm a little biased there, but I do think the time is ripe for that. Although I don't anticipate in some sense, a 2025 aging pandemic that we can immediately address and go after,
But you don't think that if you get 1,000 treatments, 1,000 different targets or whatever for the dog treatment, and then that was to clearly increase the life of the dog and the health of the dog? Basically, you can see old dog, and then after treatment, substantially younger dog, and then clear indications that a dog that would normally live 7-8 years is now, one of the shorter lived dogs is now up to 10 to 12. So basically in 5 years, suddenly the death rate of dogs under treatment drops from 10% per year down to 2, right? So that would be some kind of like pretty clear, or even down to like 1%. So something where it's like, where it's 10 times less, where basically like the evidence would be very powerful that this is like totally transformational, right? Wouldn't that be a forcing function on the whole, wow, this is now where I can see the public opinion thing, the shifting, the basically not believing it to believing it, perhaps. Wouldn't that be something that could?
We obviously think that aspect of what we're doing is ultra important. That's why we're doing it.
Mm-hmm.
I am skeptical that the FDA would change overnight based upon any single animal experiment. And in some sense, at least in the United States, to get the therapy to the human population, I think as far as a company planning goes, you need to plan on taking it through specific indications. And the best you can do, which is good, is start getting it into indications that become increasingly prevalent in the patient population. And to be fair, I mean, you start going into multiple types of heart failure, you're getting large swaths of the population. And on the metabolic side, if you're eventually able to go into things like NASH and type 2 diabetes or morbid obesity and general obesity eventually, you're talking about numbers that are approaching 50% of the population. And so I think that is the way of doing it. That's what we're banking on and betting on as a company. And in some sense, if what you talked about happens, and there's a sea change with how the regulators look at it, great. But, we're not counting on that. And I don't think it's necessary as far as getting this to a huge amount of patients. And in some sense, we call them age-related diseases because they're not specific to a specific genetic mutation, genetic variant, subpopulation. Almost everybody gets a very similar list of age-related problems. And if we're able to address those with our gene therapies, they should be available to those patients long-term. And so we think it's a fine way of trying to get it to a large, very large swath of the population. And frankly, doing it in a way that's disciplined us to make sure that the treatment is very safe and then at least is efficacious for one very particular indication.
In terms of the addressable market for the dog and the pets area, how many dogs are there, like pet dogs?
It's like 90 million dogs in the US right now.
Right. And then worldwide, probably like 5 times that or something like that, or
I think it's something like, it's like 3 times that or something like that. We are like by far have the most dogs of any country in the world. I think Europe has about I'm gonna get in trouble for guessing, but it's something like 50% amount of dogs in Europe, something like that.
Okay. And that's the healthcare market for dogs and pets is about what, 60 billion or somewhere in that range, or in terms of like how much the pet parents spend on the health of their dogs?
Well, I'm trying to remember what like healthcare spend on dogs is. I'm sorry, I'm blanking off the top of my head. I do know that, for instance, like the leading animal health company Zoetis has had a market cap like approaching 90 billion at times. And there's about 5 leading animal health companies out there. So, I mean, you can kind of get the sense for like how large of an industry is. It's incredibly robust.
Okay. Let's see. So you're not thinking that you'll need to do gene therapy, that basically the adding the gene, sorry, not gene therapy, but the gene editing part. You're doing gene therapy, and then you're doing we'll do epigenetic modifications, right? But you don't think you'll need to do actual editing of the gene within cells to get
Not
to
currently. Yeah, it's obviously really compelling tool. I find the stuff coming out of CRISPR labs like really, really interesting, but it's not a tool that I think fits with the mission we currently have. So I think, and if you look at some of the gene editing companies, they're typically looking at like these monogenic diseases with like a point mutation that they're going in and trying to change. So that doesn't really fit with what we're doing. There are like CRISPR activators and CRISPR inactivators and things like that. So, you know, I won't say we'll never use CRISPR, but the specific editing portion is just not the, you know, like we said, they're all tools. Use the right tool for the right issue. It's no different than going into your garage and seeing a screw that's sticking out and trying to take a hammer and bash it back in. You know, you want to use a screwdriver, right? Same type of thing.
You'd walk through the treatment with the gene treatment that you have, or basically the end up being an injection of this virus. For the modification of the epigenetics, would it be the same kind of thing where basically it'd be an injection as well, that you would have some kind of intervention that would go into? Can you describe how that might, you see that working?
Yeah, I'll be a little vague here, but the similar idea where you're utilizing gene therapy is the transfer mechanism. I think the major difference, and actually Noah was on a paper where this was demonstrated, proof of concept, is important to have what they call an inducible promoter on the gene, meaning when you inject the gene therapy, nothing happens. It's inert, basically. And then in presence of an inducer molecule, so you'd like take a pill, for instance, and then that would turn the gene therapy on. And so the cool part about that is it allows you to use factors that maybe you don't want what they call constitutively on, so always on, but you could basically have, you could pulse it on, and you could tune the expression by how much of the inducer you took as well. And so I think that is the major difference from what we see from our initial gene therapies to our second.
Okay. Another question I have is that leaving the anti-aging world, unless you had any comments you want to make on that. I had a question about as an entrepreneur, just a question about success with that.
Sure.
Do you have anything that you wanted to cover that I've missed in regards to?
I don't think so. You know, I just, I guess to sum up what we're up to, you know, we view aging and age-related disease as caused by a change in gene expression. We're looking to affect those gene expression profiles initially by targeting 2 specific proteins, utilizing gene therapy, and then long-term targeting entire gene expression profiles utilizing epigenetic reprogramming. We view animal health as a really key part of our strategy as we developed therapies in humans and animals in parallel. As the animal health portion both gives us really strong proof of concept data for our human programs, fulfills our preclinical data requirements, and then also long-term answers these larger questions as we're solving major challenges in the animal health market. Yeah.
Okay, that's a great work, and very interested in hoping to see progress and to talk more about that. I had a question though about for entrepreneurs. You have been an entrepreneur. You had successful exit before in a different technology from the current biotech work that you're doing. So in terms of advice that you would give to someone else working on a new technological startup, so you have the idea, you have the plan, you think that it will create this increment or transformative benefit. In terms of like getting to a next level thing, in terms of like certain key things to do, in terms of like somehow you know, getting so that you're credible enough with partners, with other things, like before to get to the funding level, kind of like certain key things to be more complete. But you say, okay, I need to work on, you know, getting board of advisors or to get other things. What would you kind of like think as one of the key aspects in terms of like going from a plan, some partners, to something that's going to be fundable, that's going to actually start being credible with people making that kind of like leap?
Well, everybody needs to get a Harvard MBA. No, I'm joking. Like, I think actually like the biggest key thing, and this sounds super trivial, but it's really not, is I I come out of the world where I've mostly focused on science-based businesses. And so I don't know, like IT or the next app or whatever, just not my wheelhouse. So if you're doing that type of stuff, maybe disregard this advice. But for science-based businesses, I think the most important thing you can do early on is really understand not the technology you're developing, the problem you are solving. And so many technologies I see are quote unquote cool. And like, I have the same instinct. I'm an engineer. I hear about a new capability and I think it's cool. I think it's intriguing. To create a company that's going to pull in the advisors, going to pull in the partners, going to pull in the people that want to work on it, going to pull in the investors. It comes down far more about what problem you're solving than how you're solving it. And so the more you can become an expert, because typically what I'm talking to is postdocs or PhDs or even professors who have spent their entire life in a specific scientific area and admittedly know far more about how this problem could be, or how they're solving things or what technology they're creating, but almost always have like minimal to almost zero knowledge on the actual challenges, the people that are currently working on those challenges, how they view the challenges, how those businesses work, how you would sell to them, what they think about this type of stuff. And so if you are looking to create a business here, the best thing you can go on LinkedIn, type in the company you think would be relevant, try to talk to anybody there and start doing that. And like, it's amazing who will just talk to you if you just reach out, particularly if you're a student or postdoc. But I can't stress enough, if you pick the right challenge to solve, how, I won't call it easy, but how everything lines up, you can create a very robust strategic plan, tactically can execute it because you have a direction. And if you don't know where you're going, it is almost impossible to make progress, because how do you measure progress? Okay, we have a new capability, but what does it enable? Like, it's all viewed in the context of solving a challenge. And so really discipline yourself to pick a challenge, educate yourself about that challenge, and become as much of an expert on the challenge as you are about the solution. And if you do that and you're able to find something where you have really nice fit between what you're developing, what the challenge is, things become a lot more straightforward.
Okay, I think that's great advice from someone who's been there and been doing it. So,
by the way, I've done it incorrectly. So I've seen when it goes well and I've seen when it goes poorly. So yes.
Learn lessons both ways.
Yes.
Okay. Well, I guess it's about our hour. It sounds like, the work, obviously I want you to succeed, and I want to live longer and
well, we're grinding for you over here, Brian.
Okay. Well, thanks for giving me the time. And I definitely want to talk to you the next time, other things have happened. If you, get the paper published, get the INDs, anything that you're doing, I'm super interested in. I actually want to tell you that also, I gave a talk 2 years ago to a business group called the Monty Jade Asian American Association. They gave a talk in their annual thing. I talked about the future for you. And one of the 8 things that I talked about was anti-aging, specifically Rejuvenate Bio, that I thought
that's awesome.
We make a load of money with helping pets, and then to use that to transition to helping people. I think that's a powerful idea.
Well, thank you for bringing us up. Thanks for getting the word out about what we're doing. And I promise to keep you updated on all of our progress. And hopefully we'll have lots of things to share in the near future.
I'm excited to hear it. Okay.
Awesome. Thank you, Brian. Thanks for having me.
Thank you. Bye-bye.